In the previous part of the lecture, we discussed about the membrane transporters. Now we will talk about membrane channels, and closely associated with channels are the electrical properties of the cell membranes or of membranes in general. So one of the most common examples or classes of channels are the water channels, or what you call the porins or aquaporins. So aquaporins, by its name suggests, they transport water molecules. But one distinctive characteristic of aquaporins is that they can allow water, H2O, to pass through, but not ions. And including in that ions is the hydronium ion. So H3O. So how do they do that? So basically, the reason why there's a need for this selectivity is, first, we do not want to disrupt the ion gradient. Because the ion gradient, from the previous part of the lecture, we know that the ion gradient is necessary, is important in the ion gauge is important in the functionings of different cellular processes. So it allows the cellular uptake of, say, glucose using the by coupling it with the ionic gradient. So in order to avoid disrupting this gradient, the aquaporins, some channels, or other channels in general, must be very selective. And this is also because channels, unlike the transporters, they are passive transporters. They follow the electron, or rather the concentration gradient. So, one of the main differences is also the rapid passage of the molecules. For aquaporins is the water molecules. Uh, it allows the molecules to completely pass through faster. So it's like comparing a ferry with a bridge. So the ferry is your transporters, because transporter cycles between uh, uh, or rather it is a cyclic, the machinery that the machinery on how it transports materials across the membrane is cyclic. So it cycles between different conformations, open on one side, then ferrying the contents to the other side. But for the channels, it's like a bridge. So it's a two-way bridge. It follows where the concentration gradient dictates it to. And so it is much faster, because it doesn't necessarily have to cycle between different conformations whenever it transports molecules. So the molecules can just pass through like a bridge, like cars passing through the bridge. So it just makes a rapid transition. So we said before, this is impermeable to ions. So hydronium ions cannot pass through due to your selectivity filters here. Now, how does this aquaporin become selective to your water molecules? Is because of what are the residues that lines the passage here. So one of the distinctive things here is the um, the carbonyl groups of the main chain of the peptide, or rather the polypeptide. It forms very transient hydrogen bonding with the hydrogen uh, of your water molecule. And looking at this scenario here, there is here what you have here, a selectivity filter. So remember, hydrogen bonds, just a review from your general chemistry, hydrogen bonds is formed between a hydrogen that is bonded to an electronegative atom. It has to be bonded to an electronegative atom, so that because the electronegative atom will suck because it's a covalent bond. So the shared electrons are mostly on the side of the electronegative atom compared to the hydronium, or rather the hydrogen atom part of the molecule. So that's one component. The hydrogen, you have a hydrogen that is bonded to an electronegative atom. So that this side, that is far away from the oxygen atom or the electronegative atom, is practically naked it's basically a naked proton. So there's a very sparse electron here. That's the first component. The second component is you have an atom that has a lone pair. And in this case, the lone pair atom is your oxygen. So the interaction between the lone pair and the proton, almost naked proton, because again, most of the electrons in the hydrogen and oxygen band here is mostly on this side, on the side of the oxygen. So you have an almost naked proton. So it's the one that is what is causing the hydrogen bonding. So you have the positive proton of the hydrogen and the lone pair of their oxygen here. Now, one restriction about the hydrogen bond is, for example, oxygen or the water in oxygen is bonded, covalently bonded to two hydrogens, and it has two lone pairs. So it cannot form three lone pair, since it has two lone pairs, it can only form two hydrogen bonds maximum. It cannot form three hydrogen bonds. Now, this selectivity filter here, the asparagine has a side chain that has a hydrogen, uh, again, a hydrogen that can form hydrogen bond with the oxygen of the water. And you have here twin, or rather two asparagine residues. And this is specifically positioned such that it it can form exactly two hydrogen bonds with the passing oxygen. So basically, it's the one that identifies this molecule as a water molecule and not a hydronium ion. Because for a hydronium ion to become, well, basically, what exactly is a hydronium ion? A hydronium ion is water, or rather, you have an oxygen that is bonded to three hydrogen atoms. And if you have a hydrogen atom here that is bonded to your oxygen, in the case of hydronium ion, you cannot form the two hydrogen bonds with asparagine. And that is what causes the selectivity filter for aquaporins. So therefore, your aquaporins allows the passage of water molecules, only the neutral water molecules, and not the ionic hydronium ions. So H plus cannot really pass through because of this selectivity filter brought about by the two asparagine residues. So basically, that's the idea between the selectivity of these aquaporins. And then again, aside from these filters, you also have the sizes. And of course, the lining, it is lined with oxygen that forms hydrogen bonds with your water. So again, the linings, the sizes of the channel, and of course, the selectivity part allows it to select only water to pass through. So aside from aquaporins, we also have ion channels. And ion channels are also highly selective like aquaporins. So here is your potassium channel, a structure of the potassium channel. So in a similar way on how aquaporins allows the passage of water, the channel is lined by oxygen, the main chain carbonyl oxygen. The same goes for potassium channels. It's also lined with main chain carbonyl oxygen. So the main chain oxygen atoms that line your selectivity filter, they form a stack of rings that provide a series of closely spaced sites of appropriate dimensions for the coordinating the dehydrated potassium ions. So potassium has a specific size. It's much larger than your sodium ion. And the size of these passages just enough so that your potassium ions as a metal ion, it can form coordinates, exactly four coordinated bonds with the lining the surrounding oxygen, the oxygen atom rings here. So sodium ions, on the other hand, is much smaller than your potassium ions. So in order for sodium to form the coordinated bonds, with oxygen, the the uh, the pore or the channel must shrink. However, because this channel is very rigid, it maintains its shape. It's very rigid shape. The energy of dehydrated sodium ions. When we say dehydrated, there is no oxygen, there is no water on this surrounding, or there's no water surrounding your ion. because sodium is much smaller, it cannot really form because it's too small to reach the exact, because the exact diameter, the exact distances of the oxygens are just enough for potassium, but too far when it comes to sodium. That is why it maintains the selectivity between sodium and potassium. How about the other ions? Other ions, of course, from the charges itself, there is a repulsion here, and then again from the sizes. And also the ions that passes through there are dehydrated. No water molecules passes through this filter. Again, because of your this selectivity filter of rings here that forms coordinated bonds with the potassium. So that is the selectivity. Some of the this is part of the reasoning behind the selectivity of the potassium ion channels. Now, for the sodium ion channel, the same reason generally, because of course the sizes. Generally, for sodium ions, it should be much smaller than the channel because sodium is much smaller. Now, this is another view of your potassium ion. This is specifically the selectivity filter. It is a very rigid side, whereas only one potassium ion can pass through at a time. And the passage of your potassium ion is such that it forms coordinated bonds with the surrounding oxygen. And it's specifically tailored for potassium and not for other ions. So this is your potassium ion channel. Now, aside from its selectivity, another way to regulate channels is by whether it's open or closed. So your ion channel, it can fluctuate between two conformations. You have the closed conformation and the open conformation. So what causes the channel to close or open? It depends on the stimulus. So there are three types of stimuli to open or close your ion channels. Although generally the idea is the channel is closed unless a stimulus opens it. So we have three known stimuli. So you have the voltage-gated channels, ligand-gated channels, whether it's an extracellular ligand or an intracellular ligand, and a mechanically gated channels. So for the voltage-gated channel, what triggers the opening or closing of this channel is the changes in the membrane potential. So it senses the difference in the membrane potential. For example, if it's a positive, there is a greater positive on the outside. The inside it's closed, but when it reverses, the channel opens. For ligand gated channels, it requires the binding of a ligand that will allow the channel to open. Mostly neurotransmitters are the ligands that opens the channel, because especially for neurons are very reliant on the electrical potential of membranes, and what triggers those electrical potentials, not only the voltage differences, but initial trigger is usually the ligands, the binding of the neurotransmitters. And then we have, lastly, the mechanically gated channels. So they are sensitive to deformations in the membrane. Sometimes you will see them in sensor neurons. When you, for example, your finger senses something, so it forms a deformation in the cells, and then your ion channel will open, which means there's a stimulus that happened. So moving on to the action potentials. So we talk about the ion channels, and because most channels again or rather ion channels allows the passage of ions, it has a great effect on the membrane potentials. So here is the voltage gated sodium channel. So we discussed previously that you have voltage gated channels, which these are ion channels that open, that are sensitive to differences in the membrane potential, and they open when the membrane potential is switched. So how do they sense it? So you have this central channel that's built from a single polypeptide chain. You have 4 homologous domains. So these domains, you have first domain, second domain, third and fourth homologous domains. Now, each of these domain has 2 transmembrane helices, shown here green, that surrounds your central ion conducting pore. So when it's assembled, you have here the conducting pore, the central channel, which correspond to the color greens here. And then you have a separated sequences. So you have the selectivity filter, which is basically not alpha helices. Mostly these are coils. So they form the selectivity filter. And lastly, you have the voltage sensors, the gray and the red. Specifically the red ones, which are used as a voltage sensors. So the red helices, the S4, it's called the S4, 1, 2, 3, 4. So subunit 4, 1, 2, 3, 4, subunit 4, because it's in the fourth subunit. It's the fourth helix of the domain. So they contain positively charged arginine residues. So arginine is positively charged, and an inactivation gate that is part of the flexible loop containing the third and fourth domain, they act as a plug. So additional domain, you have an inactivation gate as well. They act as a plug to obstruct the pore in the channel's inactive state. So basically, here comes the third conformation of the ion channel. You have the open, you have the closed, and then you now have your inactive phase. In the inactive phase, the channel is plugged. You cannot activate it even if there is a voltage difference. It remains closed because of the voltage difference. Now, what causes your opening? So how does this work? So you have your at rest it's generally closed. So your sodium ion channel here is closed. This is the resting state. So you have your negative membrane potential. So as you can see, the arginine residues, the abundant arginine residues here on this side. So it senses a positively charged exterior and a negatively charged interior. So your membrane potential is a negative. And then when it becomes open, you will see here that the potential becomes flipped. So from positive, it becomes negative. The exterior becomes negative, or rather the exterior becomes more negative than the interior. So that is sensed by the arginine-rich residues of the red sensing filters. So because the opposite charges attract. So as you can see here, the this domain, or rather this helix, the red helices, actually moves towards the exterior. And in doing so, it induces a conformational change, opening your membrane channel. And now once it opens, your ions can now pass through. Now the third state is the inactivated state. In the inactivated state, even if you have a membrane potential here, shown here, that is a positive membrane potential, basically more negative outside, positive inside. It's a positive membrane potential. Even though it is open, generally the conformation is for open, because of the plug here, it's inactivated. So no amount of membrane stimulus can induce this channel to conduct or allow the passage of electricity. And this is very important to allow the passage of our rather, this is very important in neurons, because for the neurons, there must only be one way of the action potential must only move in one direction. It should not go back from the dendrites to the axons, not go back from the axon to the dendrites. And that is how the membrane potential is propagated one way, by inactivating the voltage sensitive channels, so that even if you have your the membrane potential tells it to be open, because it's inactivated, you have a plug here, it will not allow the passage of ions. The inactivation of the membrane channel is actually the reason why action potentials are propagated in a single direction. So consider your the axon of a neuron, for instance. So your action potential is propagated from the dendrites to the axonal arm. Now you have voltage gated membrane channels here. So the first voltage gated channel will sense the membrane difference, or a charge difference, an electrical potential difference in the membrane, and it will open. So the opening will then further exacerbate the membrane potential. It will further propagate the change in the membranes. So since ions diffuse in two directions, so it can go from here and forwards. So going forward, it will the change in the ion concentration, because again of diffusion of the ions, will be sensed by the adjacent voltage gated ion channel, and it will also be triggered open. Now, when it opens, by at that the time that the adjacent or this second ion channel opens, that is the time where the previous voltage gated channel becomes inactive. Now, because it is inactive, even if there's a diffusion of ions in the because of the opening of this adjacent or the second voltage gated channel, even though because of the ionic diffusion, we will have a triggering an open a membrane potential that will trigger the voltage 1 to open again. Because it's inactivated, it will not allow the passage of ions. Therefore, what will open is the next in line, the voltage gated channel next in line, adjacent to voltage 2, this voltage 3. And so it will not affect voltage 1 because this one is already inactive, but it will affect voltage 3. So it allows the propagation of the electron potential on only one direction. So that is why you have inactive state. Aside from the open, the closed, you have an inactive state. The inactive state, even if there's a membrane potential change that would normally trigger the opening, because it is plugged. As you can see, the inactive state is plugged. Ions cannot pass through, so it remains inactive. So this is another way to look at this one. So this is the polarized passage. So the resting membrane potential, so you have here, because of the open channels, you have here a negative within the vicinity, because again, this is diffusion, ionic diffusion. So within the vicinity only of the membrane. So you have mostly negative ions on this side. Now, the change in the propagation, the negative, will be sensed by this ionic channels next in line, so they becomes depolarized. So it will open and close. But it will not go back here, even if the direction of the diffusion is, it goes both direction, because the channel ions here are inactive. So it remains such that the polarization or the action potential goes only one way, and it is the activity of the opening channel opening is restricted to those on this side, on the axonal side of your membrane, or rather of your cell. So this is how your action potential is allowed to propagate on only one direction. Now, the second type of ion channels are the ligand-gated ion channels, or sometimes it's these transmitter-gated ion channels. So at the synapse, we cannot really have voltage-gated channels. It's or rather the how the action potential is transmitted from one neuron to the other is not through voltage gated ion channels, because of the synapse. You have a space here, and that space is really large enough that simple diffusion of ions to be sensed by voltage gated channels on the receiving cell it's not enough to trigger the opening for voltage gated ions in the receiving side. So you have a large enough distance. And what is the solution here is using neurotransmitters. So at the terminal axon of your presynaptic cell, you have here neurotransmitters ready or on standby, ready to be released to the next, to the synaptic cleft, the space here between the two neurons, for easy, basically for transmitting the signal from one cell to the other. So you have, once it reaches the terminal, it will trigger the opening or the vesicle. The synaptic vesicles will then fuse with the membrane and it will be released in the synaptic cleft. So the neurotransmitter will then bind to the ligand-gated channels. So these are ligand-gated channels that are triggered to open when your neurotransmitter binds to them. So you have this ligand-gated channels. Now, the released neurotransmitter then binds and opens the ligand-gated channels. And these are ion channels. So, of course, it will trigger an action potential by itself. And then it will, what carries on the signal across the cell is the voltage-gated ion channels. So, here is the system of ion channels at the neuromuscular junction. So this is the neuron and this is the muscle cells. So the gated ion channels are essential for the stimulation of muscle contraction by your nerve impulse. So we have different ion channels numbered on how they are activated. So you have here, first of all, the nerve impulse triggers the opening of your calcium channels. And then the calcium channel will trigger the nerve impulse will also trigger the release of the acetylcholine. So the acetylcholine will bind to the ligand-gated acetylcholine receptor, which are the sodium channels. It will open the sodium channels, and the change in the voltage because of the influx of the sodium channels, so it becomes more positive now, the interior. It will then be sensed by the sodium-gated channels here and also here. So these are voltage gated channels that will sense the difference, the change in the electrical potential because of the opening of the ligand gated sodium channel. Now this fourth channel number 4, sodium ion channel number 4, will then trigger the release of calcium, which are necessary for the muscle contraction, because calcium ions are basically necessary for the muscle fibers. The sliding filament models, the calcium ions play a role in the sliding filament models of the calcium channels. And so that's how how channels play a role in electrical membrane potentials and by basically membrane potentials or bioelectrical membrane potentials. Okay, so reference books for this lecture is Albert's Molecular Biology of the Cell and also Bowett's Biochemistry.